Welcome to the Neuroveda podcast for complex health. We feature expert guests who dive into ancient and cutting edge understandings of health and disease. These conversations are about food, lifestyle, and medicine just as much as about nature, culture, and politics. I'm Jillian Ehrlich, family nurse practitioner, certified in Ayurveda and functional medicine, coming to you from our clinic, Neuroveda Health, here in Seattle, Washington. And I'm pretty excited about today's guest. Dr. Hemal Patel is a tenured professor and vice chair for development and advancement in the Department of Anesthesiology at the University of California, San Diego. And he carries a dual appointment as a VA research career scientist and a VA research pharmacologist at the VA San Diego healthcare system. He's also the chief advisor for Versia Discovery. He obtained his PhD in pharmacology and toxicology from the Medical College of Wisconsin and did postdoctoral work at UCSD in the Department of Pharmacology under Paul Insel, 2002 to 2005. And he's made some seminal contributions in shaping new investigation areas on defining the impact of membrane structure on cell, organ, and organismal physiology and metabolism, with implications for cardiac ischemia, reperfusion injury, heart failure, diabetic cardiomyopathy, aging, pulmonary hypertension, cancer, and neurodegeneration. His focus on energetics and cell biology touches upon all of these and is a control point for managing and empowering human health and resilience. Okay, Dr. Patel, welcome. We're so happy to have you.
Good to be here.
Let's start. So many questions. So let's start right with talk to us about energetics and cell biology. What is that? What are we talking about here, and why are we talking about it?
Yeah, I mean, metabolism, how the cell consumes chemicals, creates energy is the key to life, right? I mean, if we don't create energy, we don't live, we can't move, we can't dynamically create structure. And so, really, what we focus a lot in the lab at is the basic unit of life, which is the cell, and how it manages an environment that it has to maintain itself in, and how it manages that external versus that internal environment. A lot of it for us starts at the membrane, right? This defines what this little unit of life is. And then this basically creates a bag of biochemistry that then does really cool stuff. And interestingly, when you look at it, just with your naked eye, you don't see much. But when you start zooming in with a microscope, and when you start using things like really powerful microscopes like the electron microscope, you start seeing that there is this crazy organized structure that a cell has. It is not a haphazard bag of biochemistry.
Mhm.
There's a purpose to where everything is, how it's placed and how it functions and how it moves and does things. And so we've been studying that for 20 years, trying to figure out how we can hack this for health resilience.
And so as you know, my background is in Ayurvedic medicine, and Ayurvedic medicine talks about all of this. So Ayurveda really recognizes, my teacher, Dr. Ladd, used to say, every cell is the center of awareness. There is an element of intelligence that organizes everything, even within each individual cell.
Yeah. Yeah, this has been sort of, I was driving into work today and I catalog sometimes things that you ruminate on. And so one of the things I was ruminating on is one of my project scientists and I were talking last week about how we should really think about grants that weren't funded and see if we could recycle some of these. And so
Mhm.
we had this crazy idea a few years ago that there is the membrane has some resiliency to it and has a life, right? And it's not just this unique mix of proteins and lipids that just kind of exist, that there is a dynamic feature to it. And so the thought was that could membranes actually oscillate? Could they create some lipid dynamic aspects that then tie into circadian biology and how well things live and how well things die and all of those other things? It turns out that it's really hard to look at oscillations of membranes and lipids and these characteristics. But one of the things that I think we tend to ignore in life is that we live in a circadian world. The sun goes up, sun comes down. We have to live in that metric. And when you don't, a lot of your metabolism and things go out of whack and become unbalanced. This is stuff that we've been doing with Sachin Panda for a number of years now, is looking at this idea of circadian biology and clocks that we have. And I think membranes could be one of these. It's been undiscovered. And so I had some sort of cool ideas around how to actually probe this and see if they're oscillating and if they're changing and morphing in unique kinds of ways. But then really it's this idea, can the cell become this barometer of what's happening in the rest of your body? Where I think complexity happens is we're not a single cell, right? We're trillions of cells that have lots of unique structural features based on whether it's on our skin and our heart and our neurons, in our brain, in our eyes. Everything has a different shape and a functionality, and everything has a different metabolism. And so it's to try to figure out how all of that lives in concert. And I think one of the reductionist things that scientists like myself do is we try to create a perfect system to do this in. Turns out there's no perfect system, right? And so we have to learn from things and then start applying them in unique kinds of ways, which is what we spent a lot of time trying to figure out in the laboratory.
And so when you talk about oscillation, I know what the word oscillation means, but what does that mean in relation to a membrane?
So, the membrane has a lipid bilayer. And so there's a polar head and a fatty tail. And so these form this, if you were to just throw them into a mix, they would organize in that way where you'd have the things that don't like water and the things that like water that form this structural end. We think that not all membranes are created equal. So the main thought is that people think that membranes just have this bulk sort of lipid composition, and then there's just randomness of things coming together that collide and create signaling and other things. It was a good first model of how membranes, I think, work. But when we think about the efficiency of living systems, it doesn't explain how quick things are. And so when I came to UCSD, I had sort of two branching points of things to work on. This was the time of pharmacogenomics. And so there's a revolution happening and the genome was being sequenced. And so the thought was if we knew all our genes, we would know everything about how to cure every disease and how to live forever and all these things. Turns out that was a bust, right? We know our genes and we haven't figured this out yet. And so the idea that your pharmacology, things you put into your body, are reflective of mutations you have. Some allow you to process drugs in unique kinds of ways is still an interesting area to focus on. The second option I had was to move away from that genomic revolution to more cell biology. And, looking back now 25 years, I'm glad I chose this cell biology route because it's led to some really cool insights. And we were focusing on this concept that there were microdomains of the membrane, right? That there were unique structures of the membrane that were separate from the bulk membrane that did stuff. And so one domain that we fixated on was called the lipid raft, and a subspecialization of this lipid raft was called a caveoli. So scientists are not super creative in some ways, right? Naming is one of them. And so when George Palade looked at these in the 90s, they looked like little caves on a plasma membrane. So he called them caveoli, which is amazing, right? He's the father of modern cell biology, won the Nobel Prize in the 70s for describing a ribosome. And maybe that's a creative way to describe what this thing was. But they basically form these invaginated structures. And what allows for this has been a conundrum. We don't know why they invaginate. Probably some tension and pull on the cytoskeleton in the cell. But now it creates this protected environment in the membrane where outside of the bulk stuff, you have this little cave that then houses an enormous amount of lipids. So it's highly enriched in glycosphingolipids and cholesterol. But then what that lipid environment allows for is proteins to aggregate. So you have hundreds of proteins that now sit in this very small space on the plasma membrane. So now if you think about signaling from a membrane perspective, ligand comes in, a drug, a endogenous thing that your body's generating, it finds its source, right? It goes into this cave and everything it needs is right there. The receptor, the effector molecule, the downstream second messenger signaling. And so you have this efficiency. And so it turns out that this thing gets co-opted in lots of things. So cancer is one of these far extremes that we've been looking at. And so cancer makes a lot of these caveoli as a way to survive stress. If you can create more of these structures, you can keep dividing and growing and using resources and suck them away from everything else around you. We then thought about it from the other perspective. So what if we look at terminally differentiated cells like the cardiac myocyte in the heart and your neurons, right? And so these are two cells that typically when you're born with them, those are what you're going to have for the rest of your life. There's a little bit of biogenesis that happens in the heart and the brain, but not at a level where if you had a stroke or a heart attack, you're going to replace all that tissue. And so the key really becomes how do you create resiliency for an entire lifetime in these organs that are terminally differentiated? And so we did simplistic experiments where you can knock proteins out, make more proteins, and we made more of these proteins specifically in the heart and in the brain. And it turns out you make more of this caveolin protein, which drives the structural formation of this lipid raft in the plasma membrane. And these animals, when you subject them to a heart attack, their heart doesn't die. When you put them in a failure model, the heart doesn't fail.
Oh.
When you age them to 2 years of age, their heart still functions like they're teenagers, like 3-month-old mice. Same phenotype in the brain. When you express more of this protein in the brain, these animals can cognitively function like young adults in old age. And so, you learn a lot of stuff in biology. So we published all this stuff in the heart. And when you publish it, it becomes part of the public domain and you can no longer patent and do anything with it. Turns out if you can't patent it, companies are not interested because they can't make any money on it. So we learned our
Mhm.
lesson. So we patented everything on the neuro side. And so that's actually moving into a clinical trial now for ALS, which is a mitochondrial driven disease that we think starts at the membrane. And if you fix the membrane, all of the signaling and everything comes back, but then it fixes the energetics in that cell. So it's really this I think this lifelong battle that we have is how do we balance signaling? That's a huge part of what the cell does with the energetic output and input that the cell needs to survive. And it's complicated, right? It's a long process that has to be evaluated in unique settings. And then you have this worrisome aspect of aging where things start to degrade. And we think one of the first things that degrades with aging is the membrane. And this becomes this hallmark feature of a lot of the lack of longevity and other things we see. So it's been this interesting exploration of how this dynamic feature of the cell, this membrane, interacts and engages with the biochemistry and metabolism inside the cell.
Fascinating. And so the oscillation that you're talking about, is that
So what we think the oscillation does is that these microdomains aren't static. They move around. They actually move from one place to another. And so we think that and we did this. So one of the things we looked at was just gene changes in young and old animals in the heart. And so we have these animals that are negative for this protein and we have animals that are positive for this protein. And so the idea was, is this the holy grail, right? These hearts don't age. So if we look at a young and an old, maybe we'll find the answer. Well, when we did this and we looked at the young negative to the old negative, there are about 2,000 things that change in the transcriptome. DNA making RNA making protein. You look at the RNA as a surrogate to what proteins will eventually sort of express. When we looked at the two animals that were adapted, the young and the old overexpressor, and we looked at this dynamic shift, we saw 2,500 things change. So it turns out there are things that change as a consequence of aging that have nothing to do with the adaptive aging that we see in this animal that's resilient. So then what we did is we took aging out of the dynamic, right? So we just looked at the old negative compared to the old positive. And so now all the aging related things fall out and we're only looking at what adapts the overexpressing animal to the underexpressing animal. What we saw was a heavy enrichment of genes that track with circadian biology. And so
Mm.
this is what got us thinking about, is there an oscillation where the membrane, because what we fixed in this animal is just the membrane. We tagged one protein to the plasma membrane that changes the dynamic nature of this thing. And so the hypothesis of this grant that didn't get funded, which we're going to rework, was that it is really these oscillations of lipids in the membrane where the lipids are moving and concertedly changing over time in the day that circadian biology and I don't know. I mean, one of the things we have is it really circadian? Does it happen in a 12-hour cycle or is there a shorter cycle? And is there a ultradian rhythm that happens in this? And we think that that rhythm then creates this resiliency in a system. Once you lose that ability to create this rhythmic change and flow in lipids, you become static and then the lipids can get attacked and they dissolve and create all this barrier dysfunction and other things. And so we really think that oscillations may be a feature of this. How do you activate this? I don't know. I mean, can you put in a magic lipid that does this? We know that this protein caveolin can target some of these things, but then are there ways to then supplement with specific lipids that could potentially keep your membranes more resilient and dynamic? And this is something we're after.
fascinating. Well, I mean, part of it's so interesting because I mean, there's umpteen studies that show that addressing circadian rhythm or even shift workers,
Yeah.
that will change health outcomes. And so here's a really kind of fundamental way that we might be
Yeah.
reaching.
And that's, I mean, and it sort of gets to this basic nature, right? Can the membrane really be this first responder to everything that we're looking at? And I think it is the first barrier that responds to signals. And we tend to myopically think it's the nucleus. And so we did this crazy survey on campus like 20 years ago because I needed a slide for a talk I was giving. And I wanted to see what the bias was. And I don't know if the bias has changed. We should do an updated survey. So we basically took a picture of the cell And I had people in my lab walk around campus and ask people, what's the most important part of a cell? And 64% said the nucleus, because that's what we're taught to think, that the control center, the brain, the DNA is the most important element. 20% said the mitochondria. And I was shocked. Only 7% said the membrane was the most important part of the cell. And as a cell biologist that focuses on the membrane, you don't have a cell unless you have the membrane. 100% of people should have said the membrane, right? And so it's this notion that we don't think about this. When you ask people about organ systems, what's the most important organ? Skin's the most important organ, right? I mean, it's the biggest thing that covers us, and it's the barrier. And so barriers become this huge conduit, I think, to resiliency. We don't think as much about them as we should.
Yes, I totally agree. I totally agree. And we had another podcast we did, and I'm trying to remember his name, and I apologize that I don't remember his name, but he was a glycocalyx researcher. And it was a really interesting conversation because the glycocalyx is the sugar moiety that's in relationship with essentially the fat moiety, the fat components that are the lipid membrane, right?
Yeah.
And the glycocalyx, he talked a lot about how glycocalyx does a lot of our identification. So who are we when we look at identification of self? That glycocalyx carries a lot of those signatures. Is that right?
Yeah. Yeah. I mean, and it's this fuzzy, crazy thing that when you look under a microscope, a layer that sort of sits on cells, right? It's amazing.
And it brings me back to this other Ayurvedic component that I think about a lot, which is in Ayurveda, we are essentially spirit or energy first, and then we are structure second. And so when we look at what I love looking at in membranes and glycocalyx is that those are the places where we communicate. So when we think about what percentage are we structure and what percentage are we space, technically we're more, I think, space. Isn't that right?
Yeah, I would think, right? I mean, it's a bag of stuff and there's a lot of space in between all that stuff.
Although there's also the cytoskeleton of the cell, which I think when you draw a picture of a cell, there's like little organelles and then space, and that space is actually filled with cytoskeleton and
It
kind
is.
of
Right.
it's
There's a huge structure to everything and how cells sort of orchestrate. I mean, the myocyte, the cardiac myocyte that we look at has a boxcar-like shape. And even under a light microscope, you can see all the striations and things in it. And it has this shape, right? And it's shaped by the cytoskeleton that keeps it sort of in that morphic aspect.
And so, from the Ayurvedic perspective, so much of us is actually communication. So much of what makes us okay or not okay is not that, the leg bone's connected to the knee bone, but the fact that all the cells, there's an agreement that everything is okay, or there's an agreement kind of amongst the cells that things are not okay.
Yeah. And I think that's something that it's a good transition into some of this MeScreen stuff that we're working on. And
Yes.
so the question becomes, what's the highway that connects all these things? And I really think it's the blood environment. So the other sort of area we're working on right now around aging, and I have a grant pending at the VA around this, is this idea of heterochronicity. And so there was these crazy experiments done back in the day at Berkeley where they took a young and an old animal and they hooked them up by the circulatory system. And it turns out that there's an exchange of humoral factors that makes that older animal a lot younger physiologically, and the young animal gets slightly older. It turns out you don't have to physically connect them. You actually take blood from a young animal and transfer those factors into an old animal, and you can create that same resiliency. And so we have this similar idea, right? These CAV3 overexpressing animals, whether it's in the heart or you have caveolin in the brain, the whole organism ages better. One of the unexpected phenotypes we discovered is the first death in a negative, like a transgene negative, which is just a wild type animal, typically happens around 18 months. And so that's comparable to like a 65, 70-year-old human, I would think. The
Mhm.
first death in an overexpressing animal where you're just tweaking one tiny protein in the heart happens at 27 months. So
Mm.
by making the pump better, you've extended lifespan almost 10 months, which is comparable to like 3 decades. And it costs you very little energetically to do this. And so the idea that you could change an aging dynamic by creating this unique feature becomes important. So not only does the heart get better, but we think that the heart then becomes this pumping organ that not only pumps blood, but starts secreting factors from the membrane that then circulate in the blood and get to the brain, to the muscle, and to the other things. And so is there an environment that this protein overexpression in a single organ creates that then we can transfer to other living systems? And I think that communication highway really is the metabolites in your blood. That heart that's now metabolizing in a different way is creating a metabolic structure that then is feeding into things that your brain is seeing, your gut's seeing, your muscles are seeing. We think that there may be exosome trafficking that happens as well. And so now that the membrane is being changed and morphed in a unique way, the heart is packaging specific membrane kinds of things that then travel to other sites in the body that then creates some resiliency. We think there's a protein component to all this as well, right? The heart is creating a unique protein signature in the blood that is now reacting with other cells. And so this is, I think, where the next frontier in all of this is can we create resiliency through this blood dynamic, right? Changing your biochemistry in a unique way and tracking that in a live way as a new vital sign. And this is what MeScreen is after. The idea is that this blood component really becomes a global capture of you biochemically, and we can take this and put it into a living system that's never seen it, see how that living system responds as a reflection of hopefully what's happening inside you to give you some insights into what your mitochondrial biology is doing.
And can you talk a little bit about going back to that membrane piece, and then we're going to dive in more
Yeah.
wholeheartedly for the rest of the episode on the me screen itself. But can you talk a little bit about when we talk about those lipid rafts and the caveolae and the glycocalyx, what are the energetics of those things, of those parts?
In terms of how they're made.
In terms of how they're made, how do they use energy? What do they do without energy? How does
Yeah,
that?
caveolin's a protein that gets made in the sort of the nuclear ER component. It gets trafficked into the Golgi. At the Golgi, it tags to cholesterol, and then cholesterol transports it to the membrane. It then forms these unique structures with cholesterol to then create this invaginated structure. So the energetics of making the structure really ties to normal cell biology and behavior. Once the structure is formed, the question becomes, well, how does it dynamically shape and shift energy? I was sitting around and, when you're a postdoc and a grad student, kind of after a long day, you're just kind of kicking back and, it's a wet campus here at UCSD, so you can drink beer and do other things. And so I think we were having a beer somewhere in
Uh-huh.
in the space. And I sort of went back in my chair like this, and we had all these micrographs, electron micrographs plastered on the wall because they're really cool. And I felt it sort of went back and I looked at one. I'm like, that's really curious. I noticed that in one of them, there was this invaginated structure of the plasma membrane, and right under the structure was a mitochondria. And it sort of dawned on me. I'm like, has anyone ever looked at the relationship of where the structure sits and whether it interacts with mitochondria on cell surfaces or not? And I looked in the literature. No one has ever made this observation. No one's ever quantified it or went in and looked. And so we started taking thousands of micrographs and we started quantifying this relationship. And so in the heart, it turns out that almost 100% of the time, if you have this invaginated structure, it is always associated with a mitochondria. And so now you have this interesting conduit where the membrane releases things and can change and morph the behavior of the mitochondria that are subsurface. And so now you have a direct way of regulating membrane to energetics in a cell. The heart cell, unfortunately, is way more complicated than one would think. Turns out it's not just one kind of mitochondria that sit in that cell. There may be 3 or more different types of mitochondria. And so the ones that sit right under the cell surface don't really make energy. Their focus and their goal in the heart cell is to signal. They're responding to the outside environment. They're taking those signals in and they're dissipating this to the rest of the cell, to the rest of the heart. And they're telling the heart, this is what I'm sensing. This is what you need to do, kind of thing. There are these interfibrillary mitochondria that are deeper into the cell, that are the energy powerhouse, is what we typically think of. So they're the ones that make all that energy. And then we've been thinking about and describing a third set of mitochondria. They're perinuclear. They sit around the nucleus of the myocyte. And we really think those are the drivers for mitophagy pathways within the cell. And so there appear to be different and they're structured very differently. The ones that are at the cell surface have a unique structural element. They connect together in different kinds of ways than interfibrillary mitochondria. The ones around the nucleus tend to be more of this weird quasi shape between functional and dysfunctional because they're going through this flux of mitophagy. And then the interfibrillary ones are the workhorses, right? They are structured and look like pearls and they create energy because the heart never stops consuming energy. And so this is something that we've been thinking about is how does the membrane engage with mitochondria? But it really depends on the cell type you're looking at. And the heart becomes this complicated mess of mitochondria to look at. Every mitochondria in a myocyte has a function and a purpose, and that's one of the concerted things. And so one of the things I think about is that, you know, does a cell, once it starts to structure, can the mitochondria shift roles, right? Could an interfibrillary mitochondria become a plasma membrane mitochondria or a peripheral nuclear mitochondria? We don't know. And so there's a lot of open questions in all of this space. But one thing I'm sure is that the energy systems tie with how the membrane behaves and responds.
Fascinating. And so the MeScreen, named for mitochondrial efficiency screen, this is a new test. And what I really appreciated about it is that it comes from the seahorse assay, this kind of classical, well known, well used, well respected assay in the literature. And now it's kind of being made available for commercial use. Is that right?
Yeah. And so a lot of things we do on campus, we have access to crazy equipment and space and time, right? And unfortunately, the world doesn't allow that. The best way to do mitochondrial functionality is to get a muscle biopsy where you
Hmm.
have someone's own tissue, their own genetics, their own mitochondria. But you have to have a whole hospital system to do this where you can go in, one of our postdocs or techs can go in, collect the sample, bring it back to the lab, and then they can spend the whole day dissecting it in crazy ways to look at the functionality of the mitochondria. There's no way to commercialize this, right? And there's no way to give the broad public access to functionality. And so what we were thinking of is there a way to miniaturize this in a way that would give us this ability to commercialize this and get everyone access to mitochondrial biology. So nothing's perfect, right? Currently in the space, there's organic acid testing where you pee into a cup and they look at a couple of metabolites to build back what your mitochondria may or may not be doing. There's tests that look at cheek swabs and stuff like that, which, your cheek turns over a couple times in a day. So I don't really know what you're looking at. And if you're only looking at enzymatic function, it's not really looking at the coupling of electrons and flow and energy and those kinds of things. And so, again, when you have this big challenge, you start inventing things. So we were involved at UCSD was one of 10 sites picked in the NASA twin study. And the site PI was Brenda Rana, who's a good friend of mine. We overlapped in postdoc world by about a week, but we stayed friends and interacted all throughout. And this is what happens, right? When you're at an academic institution, you just run into people and you start talking to them about crazy things you're doing and you work with them. And so Brenda remembered all the stuff we were doing with mitochondria. And so she brought me into the twin study, and they said they're very interested in developing a way to look at mitochondrial function, but they can't give you muscle or any other tissue. The only thing they can give you is plasma. So could you come up with a way to use plasma as a surrogate to build this test? And so I thought about this for a while, and the simple thing was, could we actually build a human on a plate so we can buy cells that represent every organ of the body? We're doing a lot of these kinds of studies in the lab. They're called adoptive transfer, where you take some fluid from something, put it onto another thing, and you see what the change in this thing is a reflection of what's happened in this fluid. And so the idea was already there. People were doing this in different labs in different spaces, and we were already experimenting with this in the lab. And so the idea was, could you think of the plasma as a global universal capture of what's happening to that individual? So it's not really directly their genetics or their mitochondria or their cells that we're looking at, but the plasma environment that they're creating, the metabolites, the exosomes, the proteins, all the stuff that's in there is a consequence of their genetics, is a consequence of what their mitochondria are doing functionally or dysfunctionally, is a consequence of their gut microbiome. It's a consequence of their happy thoughts, their sad thoughts. All of these things create chemicals. And this happy, sad thought thing becomes really interesting. We've been doing some studies with breath work in the Joe Dispenza world, and we're looking at some data right now. There's a company in Germany that has this really fancy mass spec that can just measure room air,
Huh.
and they were looking at the room air in a German movie theater when people were watching like a comedy versus a not comedy. And it turns out the room expresses completely different organic volatiles in the space when
Ah,
there's a different emotional state. And so I have to believe that when you create an emotional state, whether you're happy or sad, it's going to change your biochemistry internally as well, right? And this is a big element of this. And so now you have this unique capture that you can get. And we were able to get, Elon Musk sent a rocket up every 3 months and we got a sample back. And so we had this samples from these astronauts before flight, every 3 months, during flight for a year, and then after flight. And on this plate, we had basically cells that represented the brain, the eyes, the skin, the immune system, the heart, the liver, the kidney. And we use these specific plasma captures at unique time points during, before, after flight to then transfer that individual holistically, not picking a handful of markers, but the entire thing onto these cells, and we looked at what the mitochondrial behavior of these cells were to predict which organ would dysfunction in specific time in flight. We're able to look at skeletal muscle, immune cells, and endothelial cells, which were the most impacted in this space. So this is the underpinning of the MeScreen, right? We don't You could do Quest or LabCorp where someone could go and get a blood draw and you'd have to process this sample. It takes a lot of energy, power, time, cold storage to get all this done. What we've been able to do with MeScreen is actually miniaturize this to a tiny blood card. And so literally it's this small card that you get in a kit. You get a little lancet, you poke a finger, a couple drops of blood onto this card. The card wicks the serum into the second space. Once that card dries, you're actually shown stability past 6 months. It's temperature stable at room temperature for over 6 months. You put it into a pouch, you ship it to us. We basically punch the serum out, reanimate it in our buffer, and we run essentially a version of that NASA type assay. We can put the entire you that we capture on this card onto a muscle cell that's never seen you, and we see what the behavior of this muscle cell is from an excruciating mitochondrial perspective. The other thing that mitochondriacs get really sort of worked up about is when someone makes huge claims about mitochondrial function when they're only looking at one element of mitochondria. And so I described the myocyte, right, where this mitochondria have all these roles. They not only make energy, but they do dysfunctional things, they do functional things, they're dynamic. And so to really get a full picture of mitochondria, you need to know how they're using oxygen and moving electrons and creating energy. But you also need to know, is that energy and that movement, is it tied to a productive end where they're making ATP or a malproductive end where they're making reactive species? They're parts of the me screen, measures reactive oxygen species generation. And then the final component, which I think is really far out and maybe more esoteric, but gives you access to stuff that no other test does, is we can also look at the dynamic nature of mitochondria because we've created this green protein tagged mitochondria where when we adopt your environment onto the cell, we can actually see how the mitochondria are shaping, shifting, moving as a way to see if your biochemistry is creating this dynamic potential in mitochondria and whether it's creating resiliency in that structural network or if it's causing that network to fall apart. Mitochondria, like all things, they're old organisms that got incorporated into our cells. Organisms are social, right? They like to hang out together. And it turns out this symbiotic organism, this alien that's living inside us, likes to be connected because then they can sense and respond to stressors. If you look at every non-communicable chronic disease, one of the hallmark features at the end of that disease stage is the mitochondria become separated, right? Then they can cause dysfunction. They can no longer sense and respond like they would in concert. And so I really think this is a way to look at your mitochondria that just hasn't existed. And it's not perfect, but I think it's the best perfect we can get right now until we get to the place where you could commercially scale someone's own cells and biology.
And so the one piece I can't quite make the leap is from the plasma to, what are you reconstituting in the plasma that is essentially representative or indicative of mitochondrial activity?
So we're looking at everything. So we're looking at your exosomes, we're looking at your proteins, and we're looking at your metabolites and everything else that's in that sample. So you could go back and reductionistly look at specific elements of the TCA cycle and other things to try to get at mechanistic understanding of what's happening. But this, from a commercial standpoint, is just a bulk measure. So what is the biochemical environment you're creating? And does that support a positive or negative functionality of mitochondria?
Got it, got it,
The
got it.
environment. So it's a
Got it.
mix of everything.
Okay. And so what you have found is that So let's walk through the parts just a little bit more, kind
Yeah.
of in the names that folks will see in their test results.
Yeah.
The first part is looking at energy profile.
Yeah.
And talk to us about energy profile based on what it's made up of, the baseline respiration, mitochondrial efficiency, mitochondrial potential.
Yeah, so one of the things we wanted to see is what happens to the normal aspect of the cell when we add your biochemistry on. And so this energy profile really is a stress test. And
Okay.
so people do crazy things, right? You're supposed to eat, sleep, move, and a couple other things to be well. But it turns out we don't do those things very well. And we with a lot of supplements to support our health, or we drink a lot of caffeine. I've been off caffeine for about a week now and I feel amazing, and I'll see how long I can continue. And so one of the things we want to see is what's the environment you've created within yourself. And so one of the things that people ask me is, well, how should I prep for this test? I'm like, be the crappiest you can be that you don't have to try. And this is how you normally are. You don't want to perform for this test. We want to see in your natural setting, what is your basic mitochondrial biology? And then can we start thinking about how to layer things in to make it better if it's worse, or maintain it if it's really good? And so a lot of people try to hack tests. Well, there's no reason to hack a test to just perform for this test, right? We want to get you in the raw sense. And so do what you're doing consistently, and get a baseline measure. So
Okay.
the baseline really gives you what is your chemistry creating to up or down regulate mitochondrial functionality. What you want to be is average on this part of the test. You don't want to either stimulate your mitochondria, you don't really want to shut them down. There is a unique profile that emerges where shutting them down could be a cool thing, and we're starting to explore this, where it almost looks like this person goes into a hibernative state where under resting conditions, they shut everything down and they're truly resting kind of thing. In most cases, you want to be completely average and optimal for baseline respiration. What that would suggest is that there's nothing overstimulating in your biochemistry that's turning mitochondria on, and there's nothing that's compromising mitochondrial function from a toxin perspective and shutting them down. We tend to see unique extremes. So individuals we've tested that have long COVID tend to have a really low baseline respiration because they have that just toxicity kinds of things. People with mold and other things tend to be on the lower end as well. We
Mhm.
tend to see that biohackers are on the higher end. They actually upregulate mitochondrial functionality because they've got lots of things circulating in their body that are stimulants, right? That are over activating energy pathways. And so the baseline gives you some assessment of that component. What we then can start doing on the Seahorse system is we have this live recording that we're doing basically on the cell, and you can add specific compounds in to probe different aspects of mitochondria. And so the mitochondria has an electron transport chain which moves electrons up this gradient. What this does is it creates this hydrogen gradient, which is then used to make energy. And so there are these unique protein complexes made of hundreds of proteins that come together that then move these things around. And so we can look at specific elements of that transport chain. One of the final elements is an ATPase that makes ATP. And so the first part of the test for the stress is blocking that ATPase. When you block that ATPase, oxygen should stop moving through that system. So the system is constantly measuring oxygen and it's constantly measuring pH in real time. And so you can look at this integration. If oxygen is still moving through that system, it suggests that there's a leak. There's an inefficiency that's born of some potential structural defect or other things that are happening that allows the non-coupling aspect, right? Oxygen should stop moving because the system is stopped. And so this gives you a sense of your efficiency status of your mitochondria, which is the next marker you get in this energy profile state. The final component of the stress test is then we actually do stress the system. We make it uncoupled, where we make it work as hard as it can. So we see what the potential drive of that system is. And this gives us from a baseline to where the drive is, it gives us what the reserve you have, right? So if you were challenged and if your biochemistry was challenged to create energy, what could you possibly create? And so that gives us a sense of the drivability of that system and the load that it could potentially create. We see variances in this. Some people just can't create that load. Other people, endurance athletes, you see this crazy rise, like 70, 80% jump. And so it's a good indicator of the adaptive things that you could create from an energetic perspective. The second part of the test uses the same system. It's basically an energy, it's like an energy balance kind of thing. So we
Mhm.
see if you're making energy using mitochondrial glycolysis, the two major ways that a living system will create ATP. In most individuals, we see that there's an equal mix. So they have a flexibility, right? So they can make energy with both glycolysis and mitochondria. Certain individuals tend to shift this. Athletes tend to be more mitochondrial than glycolytic. Individuals that are energy compromised, so we've looked at fibromyalgia, we have a few patients in the database. And again, this is not a diagnostic, but
Mhm.
we have the data and we just kind of are curious and we're looking and we see that they shift to this more glycolytic mechanism over mitochondria. And then we see individuals with long COVID tend to be compromised in both aspects, right? They really can't function in unique ways. And so this energy balance really gives you a sense of where you sit from a mitochondria versus glycolytic state. The last two parts of the test, as I mentioned, moved into the dysfunctional component. So they no longer use the seahorse to do this. We've developed a fluorescence-based assay that then looks at reactive oxygen species generation under baseline and stress conditions. And then we've created that unique cell that has the green tag in the mitochondria under baseline and stress conditions. Can we look at the dynamics of how the mitochondria are shaping and shifting? So, but then by integrating these four things, you get a sense of what the stress response and the basal response of this is, what kind of energy you're using and generating. Is it functional or dysfunctional through this ROS mechanism? And then ultimately, are your networks intact? Does this suggest that you have good lipid stability and other things in your system? And you really get a very clear picture of how to sort of think about your global mitochondrial health from this.
And then you come up with your me score. So your
Yeah, you
mitochondria.
get a score. We've been playing around with this. I think it's pretty static now. We reevaluate it every few months as we get more and more people taking the test. And
Right.
it doesn't really flip much now, right? We have about 4,000 people that we've used to create these unique profiles and scores. And so there's unique drivers of this. And we see unique elements, right? If you're in this lowest tier, this low bucket, it's really structural lipid kinds of things that you're struggling with. And I really, as a cell biologist, I think the way to fix your cell really is threefold, right? That you have to fix the membrane structures because those are the first to degrade and do these crazy things. One of the things we see with just human nature is just tell me what to do and give me all of it, right? And they want to do 14, 15 things at one time. That's not how biology is wired, right? Everything is layered. You have to start building the foundation first, then move to the next thing, then move to the next thing. So everyone wants to get to this first thing. And so they'll do all of the crazy things to then unwind the structure. One of the things that most people don't realize is if you don't have an optimal structure in your membranes, taking certain supplements may actually damage that inside of the cell and the membranes even more. And it gets you even further down the line of dysfunction. And so for people who are in this low tier of a low me score, typically you start with lipid supplementation, phosphatidylcholine, serine, ethanolamine, and inositol to rebuild bulk membrane structures in your plasma membrane, your nucleus, your mitochondria. When I say lipids, most people think of omega 3s, which is more fancy lipids that you need later on to create specialized domains, but you really need bulk membrane support. Once you fix the membrane, if it doesn't fix all of the other problems that are happening, then the next sort of layer you have to start working on is mitophagy, autophagy. As we get older with chronic diseases, this is a key pathway that's targeted in biology as a way to muck the system up. And sometimes you have to do crazy interventions, time restricted eating, cold stress, heat stress, supplements like urolithin to just activate these pathways within the cell. The final step, if those two things don't fix your mitochondrial biology that we struggle with and a lot of people struggle with, is oxidant, right? We live in an oxygen rich environment. This is the other thing that we're very interested in this structure on. It turns out that there is this interesting evolutionary link between the rise of oxygen in the atmosphere with the rise of animals moving from water to terrestrial environments with the rise of cholesterol in our biomembranes. Turns out cholesterol is a sink for oxygen. It binds to oxygen and tempers and holds it. So the other hypothesis we have around this microdomain in mitochondria is that this could be a channel for oxygen into the cell, and the
Hmm.
alveoli then actually hold this in a very tight space and they deliver this oxygen in a very tempered way to mitochondria. Because if you had unopposed flow of oxygen into the mitochondria sometimes can't handle all of it and they'll make free radicals. And so by having this buffer system that creates this regulation, you never have that aspect. And then one of one of the hypotheses we have for why these animals are adapted to things that cause hypoxia, like ischemia reperfusion injury, heart failure, is that if you enrich this membrane with oxygen, even under those stress conditions, the membrane is holding on to so much oxygen that the sub mitochondria never become hypoxic, right? They have this ready-made source of energy that they can create under these toxic stressors. And so it's really this problem that we face once we sort of solve the membrane and the autophagy problem is we live in an oxygen world. It is creating molecular fire in our bodies, and we have to manage that in a unique way that limits that fire, the reactive species that are generated. And so if you don't have this activation of endogenous antioxidant systems, is to take some exogenous antioxidants or polyphenols and other things that will support pro sort of reductionist kinds of things with the free radicals. And that's it, right? I mean, it's a very simple way to do it, but probably the hardest thing for people to do is to slow hack all the way up to fixing mitochondrial health.
And so what I like about MeScreen is that it gives you all of this information. It breaks down those 4 tests that you were talking about, and then it gives you a total score, and then that score breaks down into these 17 different profiles, and it tells you where you are so you know exactly where to start.
Yeah.
Just so could do you think any of our cholesterol, like our hyperlipidemia epidemic, could be our body trying to sequester oxygen? Is that crazy?
Yeah, maybe. I mean, that's you want cholesterol in your lipids. You don't want it floating around in your blood, right? I mean, that's the big sort of question around it. Interestingly, one of the early effects of statins, first generation statins, were cataracts. And so it turns out the lens fiber in your eye has one of the highest densities of cholesterol in your body. And so if you get rid of that cholesterol, your eyes are exposed to the oxygen environment. And so that oxygen now, because there is no lipid buffer for that oxygen, creates oxidant things and creates the cataract. And so there is this interesting tie between cholesterol and cholesterol floating around in the blood and possibly this oxygen dynamics within a system.
That's really interesting. It's really interesting. Interesting. Okay. 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 So here we are. So how does if I'm thinking about that German movie theater and
Yeah.
how you can really check even within an hour of watching a comedy or non-comedy, you can change kind of the atmospheric conditions. How does Me Screen get a durable kind of not moment by moment?
So we've done morning, evening, fasting fed, and it's really not a thing that's like a glucose measure, right? It's not dynamically shifting. Your biochemistry takes time to shift. And so we really think it's a 2 to 3 month process to turn over all of those things that are then coming together in thousandfold to create changes in your mitochondrial biology and things. There are certain things that will push this very fast. We've done some ayahuasca samples, and you can get a change in mitochondrial function in 48 hours after an ayahuasca trip. We're working on
It's the same direction?
it goes positive, and then it drops back down by the end of the week.
Huh.
But it's not a sustained change, but it is something that you can dynamically shift, and it
Huh.
supports stuff that's in the literature with psychedelics and what they do to mitochondrial states and energy states as well. So there may be certain things that do this, but again, it's not sustainable, right? It can cause this rise, but you can't maintain that long term unless you keep doing ayahuasca forever, which I don't know if you want to do. Some people maybe.
I mean, if you've got real problems, you need real solutions, right?
Yeah. And I mean, we're working with a lot of companies that want to test their products and things out. And there may, a lot of them are interested in shortening that time window. And I don't know if that's really the goal, right? And there may be products that can create an effect in a week. There may be products that do this in a month. But I think the longer you give your mitochondria to adapt to a new sort of lifestyle and a change, the better resiliency you're going to get. The other aspect to this is I think it's a lot of our health is our behaviors. And a lot of things we can do really well for a week or 2 weeks, but we then struggle to go beyond that, and I'm a main culprit for this as well. I think about things and I'm like, "Okay, I'm gonna focus on this," and someone's given me this Akkermansia supplement that I'm supposed to take, and it's been sitting on my desk, haven't done it, and I'll do it for 2 weeks, and then I'll feel really great, and then I'll think about something else, and you move on, right? And it's really changing pattern behaviors for a long period of time that's gonna make dramatic changes in your health.
Have you looked at a kind of trauma therapy or hypnotherapy or other types of mental health shifts? People can have an experience like an ayahuasca event but do this in therapy over time. Do you find that those mental health changes or something like TMS can
Yeah,
change.
that's a good idea. We have not done that. I know there's people like Martin Picard and others who are in this space that are very interested in the mind mitochondrial connection. And so I think there's going to be a space for this. I think a lot of mental health disorders are metabolic in nature. Could you use mitochondria as a way to fix a lot of these?
Or I guess that is would be the Joe Dispenza word.
As we are exploring that in unique kinds of ways, we'll have to schedule a second hour to talk about all the crazy stuff we're working on in
So
mind
interested.
body space.
I really, and obviously just to put the plug back in for Ayurvedic medicine, the changes that we've seen after especially something called panchakarma can be tremendous. And then have you worked with this MeScreen and plasmapheresis?
We haven't. There was a group that wanted to work with us on this, and I don't think we've any materialized any studies around that yet.
Yeah, so that would be also very interesting because there's so much environmental, like, and by environmental I mean like plasma environment changes with plasmapheresis. And the differences between, the two types of plasmapheresis being centrifugal versus the membrane, double membrane
Yeah.
filtration. So induced pheresis. So very interesting. Fascinating, fascinating. Well, more to come. I'd love to schedule another hour, maybe in another 6 to 12 months, we can kind of review everything that's updated.
Yeah, that sounds great.
That sounds great. Thank you so much. And I want to thank you for being here today. And thank you, everybody who's joined us from home. We love that you've joined us to discuss how to make our whole world medicine. We rise or fall together and are committed to sharing knowledge and insight that empower us and our communities towards greater health. Thank you for listening to the Neuroveda podcast for complex health, and we'll see you next time.